I am Kasia Głowacka from University of Nebraska-Lincoln. Light is harvested by antenna complexes, which works like a funnels. They are very complicated structures, and the light harvesting complexes built mainly from pigments and proteins. However, we can show it work. We can show as a simple funnels which are harvesting the sun and pushing it to the processing center. However, when there is too much light, there is no enough processing power in the processing centers, and then the ROS, the reactive oxygen species, are created. What we know about the ROS, that is destroying the lipids, the proteins, and we know also that this is the agent which is making us looking older. When there is not enough light, usually there is enough processing power in photochemistry to use all the excitation of the energy. However, where there is too much sun, some of this energy is released. We can say that this is through the safety valve, that how non-photochemical quenching works. It release this energy is released safely. So when we think about the abiotic stresses, then when there is too much sun, what is happening, the photochemistry is somehow limited because of the abiotic stresses. Then there is more need to release too much energy via MPQ. So we could think that there is much more and different regulation of MPQ during the abiotic stresses. And MPQ can be quantified very easily through imaging of fluorescence. And how that works, here is the hand of my postdoc who is holding the extract of chlorophyll. When we shine the blue light through the chlorophyll, it's releasing the red light because the chlorophyll molecule from the ground state level is lifted to excitation site because of the photon of the energy which is absorbed by it. And then this excitation state can come back to the ground level when the photon is released back, what we can observe in image, and released by heat, which is MPQ, or of course can drive the photochemistry, which we hope most of the time happens. If not, then all this left part is happening. So single chlorophyll molecule, it's connected with another single chlorophyll molecule. It's created triplet, which can react with oxygen and create the oxygen reactive species. We use the fluorescence pictures, which can look like here. So usually we have black and white cameras, so they are only gray pixels, but we can assign them different colors, and then we can be more excited about this beautiful pictures. But what we can get from the fluorescence pictures is many of the very useful traits. I will be focusing on two of them: non-photochemical quenching and photosystem to operation efficiency, which is 5PS2. We abbreviate it to 5PS2. So a little bit about myself. My lab is located in Midwest of US, which is this red state here in Nebraska. And I work to improve the abiotic stresses in commodity crops which are surrounding us. So Nebraska is quite flat, and we have a lot of the corn and soybean, and generally is the land which is producing food for US. We're lucky in my lab to discover to design the semi-high throughput phenotyping assay based on the 96 well plate. So how that works, we go to the field, we collect the leaves, we put them leaf discs, we collect them, put them in the plate. We use the moist sponges to preserve the discs, and also to push them against the bottom of the plate so they don't move. And then we can collect time series of measurements which are giving us opportunity not only to measure some value of MPQ but assess the kinetics of it. So here we have the kinetics of induction of MPQ and relaxation in the dark, and also induction of 5PS2 in the dark. So when I look at this curve, I see the hyperbola. So we can use many mathematical equations to fit and assess, for example, the rate, so the speed of induction of MPQ, and also the steady state. So what will be the maximum MPQ when the kinetics is following this shape. And also in the dark, one more time, the rate, the speed of relaxation, steady state of this process, and also the residuals of MPQ. So that means the MPQ which is not relaxed in the end, for example, of 10 minutes. So that will be the MPQ which is competing with photosynthesis in the dark, but also in abiotic stresses is the residual MPQ which will be protecting in the next stage of the light phase. In my lab, we use multiple techniques to work on the regulation of MPQ and photosynthesis. This is the gas exchange, imaging. We use the high throughput phenotyping facilities in the greenhouse, but also in the field. We use transgenic plants and metabolomics. We like to think that in the core of everything is the energy of the sun, which then can go in three ways. Can drive MPQ or photosynthesis, or can be released in not regulated processes. And that is turning into photoprotection, assimilation of CO2, and photo damage. And we know that because here is the core of the energy, the redox processes are very strictly related to photosynthesis, to the light reaction of photosynthesis that can influence the gene expression, the metabolomic changes, and stomata behavior. And then that what we most like to say about the plants is how they use the resources. So that influence the photosynthetic light use efficiency and water use efficiency. So before we use our semi-high throughput phenotyping assay for MPQ, we checked if the results obtained on the leaf discs or on the whole plant agree. And here we have beautiful relation, almost 1-to-1, saying that both of the methods are showing pretty much the same results. When we extract some parameters of MPQ, we see that there is no significant difference between them. And for that, we use wild type plants, but also the mutants which don't have much MPQ, and the overexpressing lines which have more MPQ. Our next step was to just go and measure phenotype, the natural variation in MPQ and 5PS2 kinetics in some crop. We collaborate here with James Schnabel lab, also from UNL, and we look at them. Maize field. We harvest the disc in 2 years, over 800 unique maize accessions, and we combined it with the 900,000 of the SNPs. First, we saw that there is a big range and disconnection of MPQ parameters. So big range, when you look here at the green or blue line, we see that they are ending up in very different places. What is the disconnection? That although they have very high speed, one is ending up low and one is ending up high. What that means that most likely different genes are related to these two different phases of MPQ. So our next step was to look if we can find the genes related to different phases of different parameters of MPQ and 5PS2. Successfully, we accomplished that. We find out over 120 regulatory genes, and we focus on 6 of them. I'm showing 4 here. And for example, we find phytosulfokinase simulator 3, which is related to pattern of stomata and also responses to the phytohormones. This one was the most significantly associated with 5 MPQ kinetics traits. Another one was tioredoxin Y1, about which we don't know much, but here it was correlated specifically with the traits in the dark, both 5PS2 and and MPQ. Another thioredoxin, the atypical cysteine-histine-rich thioredoxin 3, was related with the biggest number of the traits, which was 9 traits, mostly MPQ. Another example, it was the transporter in outer envelope of chloroplast, and that one was related specifically associated only with 5-PS2 kinetics. For 6 genes, we look if they can also have the pattern, have the effect on Arabidopsis, so C3 and C4 plant. And we look here at the mutant in thioredoxin Y1, we see that relaxation of MPQ is affected, however, also the induction of MPQ. So we see that genes found in maize have also effect on MPQ in another species quite far away in the evolution, which is C3 plant. Another excited result from our study was that we saw very high consistency of the effect of the SNP across 2 years. So this is one of the genes which we found. We have two possible SNPs in one nucleotide, C and T, and we see that across 2020 and 2021, always, for example, the blue, so the C allele has the higher and significantly higher value for all the traits, but not oppositely we see in the slope, than the allele T. And of course, the values of the traits are different across 2 years, but very consistent effect is a great thing to see. That means that we can repeat our results. Most likely, the strong effect of genetics we'll see in another years and in other locations. Now, after finding out these regulatory genes, which is really awesome, and it's increasing the toolbox which we can use to engineer the right MPQ level in the plants, we ask if they are the same genes which we find in the control conditions are also responsible for MPQ in abiotic stresses. We focus here on the stress of the low nitrogen. It's very unique stress when we think about that because it's long-lasting and what gives a possibility to very big adaptations. So when we think about that, we can look at this funnel, which is the example is some kind of illustration of the antenna complexes with the processing centers, that these antenna complexes can get smaller to get less light harvest, or they can be built from different pieces. So in this time we also collaborated with the same lab, and we investigated 225 maize accession grown on 2 conditions, control and low nitrogen soil. We look at 3 stages, early vegetative, late vegetative, and also the post-flowering. And we saw that almost the same portion of the accessions is increasing MPQ in the stress, or keep it the same, or decrease. We focus on these 2, pink and yellow group. We went to the field, and this time we checked the MPQ on the leaf levels using different methods. This is the MPQ calculated, not measured. And we saw that both groups under control condition soil have similar MPQ. However, in the low nitrogen group A, it's upregulating MPQ, and group B is keeping the same values. That was very exciting finding. And we look at what is possible regulation of this upregulation or downregulation of MPQ, or keeping it the same. We look at the chlorophyll A and B, which obviously is a component of the harvesting antenna complexes. This is true for chlorophyll B. Chlorophyll A is in both antenna complexes and processing centers. And we saw very strong relation between chlorophyll A and B ratio and relative change in MPQ max. The group A, they are the blue signs, are behaving in such a way that they have a lower chlorophyll A to B, but higher MPQ, and group B is staying the same, no change in both of the traits. What's happening is that group B is downscaling the funnel and processing center almost to keep the same ratio between them, but group A has smaller processing center. We saw that it's less chlorophyll A, but relatively still big antenna complexes, which is causing the need to have more MPQ. In that case, we saw that the group A losing more biomass in comparison to group B. So we can say that keeping the same proportion of chlorophyll A to B, it's giving the possibility to keep the MPQ not change and keep the plants having bigger biomass. We didn't end up here. We use one more time GWAS to find the genes which are related to this regulation of the chlorophyll A to B changes and MPQ. And that was done in the sorghum in 339 sorghum genotypes grown in the field in control and low nitrogen. And out of two biggest targets, one was the no yellowing 1 gene, which is coding the magnesium dehalatase. And this is the enzyme which is involved in the first step of catabolism of chlorophyll. So we find a gene which can cause these changes, and therefore they are strictly related with MPQ. That was very excited. We look in this group of the sorghum one more time on chlorophyll A and B in two groups carrying one or another allele in the no yellowing one, and we saw that in the control conditions they don't have much change in the chlorophyll A and B. But when we grow them the low nitrogen, what is happening that the group A has lower and group T has a higher chlorophyll A and B, and this is in the greenhouse conditions. In the field, we see the same thing, that in the control conditions, no difference between them, but in the low nitrogen conditions, one group, the group A, is have more active this gene, less active, we can say, more active this gene, no yellowing one, and is causing that we have much less chlorophyll A in comparison to chlorophyll B, what is causing that more MPQ has to be regulated, more MPQ has to happen to protect the photoantennas and reaction centers. Another question which was interested for us was if MPQ under different stresses in the same genotype will be the same or not. For that, we look at this very not noticeable plant, which is Arabidopsis thaliana, but is the model plant for which there is multiple resources, and a lot of the labs are studying Arabidopsis around the world. We look at the ecotypes which were found in many locations, in different locations in Europe and in Asia and Africa, and here we have 3 of them. And what we see that in the chilling, this particular genotype is upregulating MPQ, but not in the drought. Another one is upregulated in both of the stresses. What is telling us that genotypes, there is big genotypic variation in MPQ, but also that response really is related to the stresses. So there is not that one genotype will always upregulate MPQ. There is much more regulation related to specific abiotic stresses. And that's, from one side, that's good. That means that my lab can still work on MPQ, and there is a lot of the different questions which we have to answer. As a summary, we show that for the aim of engineering tolerance or breed the tolerance in crops, and the emerging target is, for example, chlorophyll catabolism genes like no yellowing one. For the control conditions, we find another genes which are thyroidoxin and, for example, chlorophyll enveloped transporters. We saw very genotype-specific NPQ response to the low nitrogen stress, which is related to chlorophyll A and B ratio. And in early and late late vegetative stage, we see that similar portion of the variation is shown as an increase and no change or decrease of NPQ, but not in the post-flowering stage. That might be related that in the post-flowering stage, there's more degradation of NPQ, what means that this phenotype, some of the genotype will still be able to protect the chlorophyll from degradation, but some of them no, and that's why we see the decrease of NPQ. Thank you so much for your attention. This work was possible to be done because of the hard work of my team. 3 postdocs, Sima Sahai, Asha Kumari, and Ben Turk, and also graduate student Jared Haupt, cohort of undergraduate students, and collaborators. Particularly this part of the work was with collaboration with James Schnabel Lab from UNO. And this work was funded by multiple sources like EBSCO-R, Nebraska, NSF, CIRI, which is Centrum for Roots and Rhizobium Interaction, Nebraska Soybean Board, and USDA NIFA. Thank you so much.